Recording in progress.
Recording in progress. Hello, everyone.
Hello.
Hope you're having a good day. Welcome to our BMID podcast for what is this? Shigella and Salmonella. And we have here today our star faculty, Sario Hermantara. Hello, Sario.
Hello, Miss Eliz. Hello, everyone.
How are you today?
I'm okay. I'm still recovering from yesterday, from recording of the nematodes there. But yeah,
Have
we'll
you
see.
eaten any udon yet?
No, but I've dreamt angels and Pegasus that drinks blood.
Ah, cool. Did you ask them if they have tummy ache? All
No,
right,
that
let's
slipped my
move
mind
on.
though.
Okay, so tonight, if you dream of them again, can you please ask them? Because I really want to know.
Oh, sure,
Do
sure.
you think they have Shigella and Salmonella in them? The
Well,
digestive tract.
for that, I'm 80% sure that they will not have Shigella and Salmonella, especially the tippy ones, because these strains or these species are, I guess, what's the term that I'm looking? Exclusively human pathogens. How
Ah,
cool is that?
so choosy.
Yes. Yeah, not sure why though, but we'll cover the pathogenesis, I guess, the more pathogenesis, and then that might give some clues later on, right? So let's just get to it.
Yay.
Right, learning goals. Who wants to see that? Um, right. So I have here a slide on enterobacteria, which should have been covered in your class, right? And the E. coli class before.
Yeah, so fun. So wait, what's the difference then between E. coli and Shigella and Salmonella, apart from the difference in their names, and E. coli is so ostracized because their names don't rhyme with the rest of them. What, Salmonella and Shigella are sisters?
Yeah, they're not stop or strep. But before we go into the differences, probably let's talk about the similarities first. Right, so E. coli, Salmonella, Shigella, as well as Klebsiella yersenia, they can be grouped into this enterobacteria grouping, which are basically gram-negative rods. So they are all filamentous rods.
Wait, they're all rods?
Yes, they're rods. However, I did read somewhere that there are some that are cocci as well, but the important part here is they are gram negative. So this is to differentiate those that are enterococci, which are most likely gram positive bacteria.
Mm-hmm. Interesting.
And yeah, I'll just go through these points. They grow under aerobic and anaerobic, so most of them are facultative anaerobes. And they do ferment glucose and lactose as well. And that's basically one of the foundations on how we select or how we differentiate or identify bacteria. Right. And yeah, they are by far the most common cause of UTIs and also diarrhea. Still on Enterobacter, so there's this components, these virulence components for these group of bacterias, and this can be classified into three major antigens, O, K, and H, which I believe is already covered multiple times.
Yeah, so we've covered this O, K, and H antigens for E. coli. So these are the same for Shigella and Salmonella as well.
Yes,
So
yes.
how would you differentiate between E. coli, Salmonella, and Shigella then? Do they also have numbers? Like when we talk about E. coli, we give them numbers, right? So O157, H7. So Salmonella and Shigella each will have their own numbers.
Yes.
Can they have the same number as E. coli?
Now for that, I'm not too sure. However, those number corresponds to the type of antigen or the type of side chains that is added on if we're talking about LPS. For the capsules, I'm not too sure how the numbering is, but most likely it would be the type of, I guess, the building blocks for the capsules itself. As for the H is on the flagellas, I guess. Right. So yeah, they do have the same system of naming of their serotypes, similar to E. coli. Right, and also a quick review on identifications. Now, because they're quite specific and then gram-negative from the GI tract, so one of the most used identification method is growing them in McConkey agar, which selectively grow gram-negative. And also in McConkey, they add in this lactose supplement. So they can differentiate bacteria that can ferment lactose or not. And as you can see there, those are the different results, I guess, from colonies growing in these McConkey agar. Additionally, catalysis tests can also be used. So Enterobacter will generate positive results and negative results for oxidase tests.
I think they've tried this in the lab, right?
Mhm.
So in your picture there, the salmonella gives brown colonies, right, or white colonies, which means they're non-lactose fermenting.
Yes.
And the figure beside that, Staphylococcus aureus, doesn't give a colony. They don't grow on McConkey.
Yeah, they do not grow. That's a really good eye you have there, Miss Ellis. And do you know why?
Why? No, I don't.
Maybe it's a technical error.
What?
No, no. Well, it's because they are gram-positive bacteria. And yeah.
No, so the empty agar, McConkey agar is that pinkish color.
Mhm.
And when you grow the non-lactose fermenting, it turns gray. Sorry, what is this color? Cream
Reddish.
brown color. The color will change.
Yeah, there's color change there. But it should turn into like bright red. So like E. coli there.
Yeah, yeah, for lactose fermenting bacteria, it will grow bright red, right?
Hmm.
But if it's non-lactose fermenting, then it will also change color
Oh,
to
yes.
brown.
Yep. Right, so that's overview of Enterobacter, and you've already covered E. coli from last week's session. So this week we will be covering two bacteria, Shigella and Salmonella.
Shigella. Shigella is such a pretty name.
Do you know where did it get its name from?
Yeah, where did it get its name from? Wait, let me guess. We have Shiga toxin, right? Is it somebody called
Yes,
Shiga?
you are correct.
Yay!
Yes.
But why does it become Shigella? Because they like Miss Ella.
I'm not too sure about that. But what I'm sure is that Shigella is named after a Japanese physician called Kiyoshi Shiga.
Is that because Shigella is commonly found in Japan?
Most likely, yes, because the Wikipedia page here, Kiyoshi Shiga was a researcher that researched, wait, what is this? Researched the cause of dysentery,
In Japan.
obviously in Japan.
Well, not always. Maybe he did his PhD somewhere else.
Okay, let me just scroll down here. Entered Tokyo Imperial University School of Medicine, 1892. Wow.
Wow.
That's 100, more than 100 years ago. 130.
So they've been around for a really long time.
Well, yes, most likely, even before this discovery, right?
Mhm.
Well,
All right.
anyway, Shigella here. So the antigenic makeup would follow the same classifications as all the other Enterobacter, so that OKH. However, they are lacking a flagella, so they are lacking the H part. So only OK, right?
They're
Um,
always okay.
they're always okay. Yep.
What
Um,
nice guys. Wait, so if they're non-motile, how do they get around?
that's a really good question. We'll get to that in the next slides. But before that, just to make sure, we've covered this that Shigella only has 4 species. So this is what you read in the slide here. There's Shigella dysenteriae, Flexneri, boydii, and Sonei. And they are classified into serogroups of A, B, C, and D. Now, the serogroups is depending on the O antigens there. Their natural targets are enterocytes, so they are the epithelial cells and the GI tract. Before we get into the GI tract, probably just cover the epidemiology here. So Shigella are strict human pathogens. Oops.
So this is
Trans.
why they can't infect your angels and Pegasus.
Yes, because they only target humans. Nasty little creatures here. Transmission is by fecal oral route, as like almost all pathogens. Well, not almost all pathogens, but most of bacterial pathogens.
Enteropathogens, right?
Yeah, enteropathogens. Guess that's correct. Now, this is interesting because they're strict human pathogens, right? No animal reservoir. But as we can see here, there's an estimate of 450,000 infections in the US alone. That's per year. That means the invasion rate is quite high. Now, why is that? Now, usually it's high because there's animal reservoirs or there's vectors that comes to play. But in this case, it's just because these bacteria are really efficient in infecting humans. As few as 10 organisms would be enough to infect one individual.
Yeah, so I find it very interesting. So if they don't have an animal reservoir or any other organisms that house them, that means that we can only get them by exposing ourselves to fecal matter of human,
hmm hmm
right?
All, yes, all contaminated foods or water that interacts with fecal matter, I guess,
Of humans.
of humans, yes.
Yeah, so that highlights how often we actually encounter human fecal matter in even in developed countries like the United States.
Yes, yes, that's also an interesting point there. I
So
guess it's
even if you don't scratch somebody's anus and suck your fingers after, you can still so they might have they must have very good survival mechanism or survival rate on things then.
mhm,
Because if they require, fresh fecal matter to be transmitted to somebody's mouth, then the infection rate will be a lot lower than that. So they must have a way to survive very well in their environment to wait for somebody to arrive in somebody else's gut tract, right?
mhm, mhm. Well, yeah. No, I'm not too sure on how well they survive in the environment, but what I can imagine is that for example, like in one household, it's going to be easy enough for the transmission of these bacteria from one person to another, just because, well, they only take like 10 organisms to infect. And and that would be what is that? The epicenter of infections there. And then the I guess the how well the sanitation is in that house or in the neighboring house or in the neighboring apartments and so on and so on will have a huge effect on the transmissions of these bacteria. But really good point there. Mostly because the water is being recycled every time.
Yeah, might be. Would be nice to know how to get rid of them or how to sanitize food and water. Um, Pretty sure there will be some recommendation on how long and to what temperature you should heat your food and stuff like that to be able to kill them.
Yes. No, I did not put that inside the slides. But most likely, the general requirements for heating up food, cooking water, and so on, would be enough to kill or repel this
Mm.
Shigella
With
pathogen.
parasites, you get parasites like worms and stuff like that by consuming, um, for example, meat infected with the parasites, and then you don't cook them well enough, so their inactive forms can survive. But how you can kill them in the meat or in the vegetation is by freezing the meat and thawing it again. I wonder if that will work as well with bacteria.
Mm.
Because with parasites, with worms, once you freeze them for some time and then you thaw them again, they would have died. But I think bacteria probably is a bit more hardier than that.
Most likely, capsule-forming bacteria will withstand that. Um, but
So
for
the
these
Shigella is not
organisms,
capsule forming, right?
no, no.
They don't even move around.
Yes.
Okay.
So they're like these, what is this? Chicky balls. No, not balls.
Yeah,
They're like
looks so
those.
yummy. Like chicky balls. Those yeah, I don't
Yeah.
know what they're called. Cheese sticks,
Yeah,
giant
balado sticks.
fingers.
Balado sticks.
Yummy.
Okay, so now we move into how they infect our GI tract. So, but before that, just to make sure everyone remembers the cells that make up our GI tract, here is a nice figure here. So as you can see, the lining of the GI tract is made up by these enterocytes, which is these orange colored cells. Now, in between the enterocytes, there are these M cells, these green layered cells that look like a bit funky there, that concave shape there. And these M cells are called as microfold cells. They're specialized epithelial cells that is basically shaped that way because they want to sample bacterias inside the intestines. Now, sampling bacteria means that they will also need to not only sample but also analyze the bacteria itself. In this case, it's the antigens of the bacteria, and that would mean exposing these antigens to the immune system. And the immune system in our GI tract is basically uh, what is this? Not mediated, but made up by these follicles, uh, which is which couldn't be called as this gut-associated lymphoid tissues. Is that correct? Eliz,
Yeah,
help me out here.
sure. Yeah, so specialized lymphoid tissues that guard the gut lining, right, on of gut from gut pathogens. And these M cells are actually similar to macrophages, right? So they um the way they sample the antigens, as you described, would be by endocytosis or like phagocytosis of those gut bacteria, whatever it is, and then churning it down to individual antigens to then get passed on to our dendritic cells.
Hmm. Yes, yes.
Mhm.
They also have specific receptors, I guess, for these types of bacterias. And for normal microbial common cell flora there, that would not be too much of a problem. But for pathogens or I guess bacteria that's coming from outside, this would be the screening place, I guess, for the intestines, just to make sure that there are no alien bacterias invading our tissues. And yeah, as we can see in the figure to the right there, that's much more looking closely in these M cells. And yeah, as you can see, there are these different types of receptors which recepted all the bacterias, and then the bacteria get phagocytosed and processed for antigen presenting to the dendritic cells. And dendritic cells will present that to the B cells, T cells, macrophages that resides in the gut. Now we look into what will happen if there are Shigella inside our intestines. So first off, Shigella um, is acid resistant, so it will survive the stomach acids there. And yeah, I guess this is probably the important one. So fundamental pathogenic event of Shigella as well as Salmonella later on is invasion and destruction of the human colonic mucosa. They do have slightly different ways though. Right, so first off, Shigella needs to attach. And now, right, so let me introduce to you our little Shigella here, which we call Sig. Right, hello Sig.
Hello, little cute shig.
Do you know what Shig is short for?
Shigella.
Well,
Anything
Shigella,
else?
Shigella is the name of the group bacteria, but
Uh-huh.
Shig is short for Shigit. But this little Shig wants to be called as Shig. Okay, so let's just call it Shig. Right, so first, Shig will invade M cells because it recognizes the receptors and on the M cells itself. Now, what it does is like it gets engulfed and then it transcytoses, meaning that it will uh, move from one place to another. I forgot the term there, the intestinal
The
tract.
lumen,
Yes, the lumen to the
basal side, the
lamina. Yeah,
basal
basal
membrane. Yeah,
lamina. Yes, yes. Thank you.
you're
But yeah,
welcome.
basically transcytose to the basal lamina, which, as we see in before here, has all those macrophages, all those white blood cells. So natural outcomes would be invading these white blood cells, or in this case, macrophages. And this little Shig is able to do so and induce apoptosis in the macrophages itself. So it does not replicate in macrophages. Okay, so probably that's one thing to underline. But it kills macrophages.
But it's probably because they just move and then they got lost inside macrophage, right? I mean, if they don't replicate that, it's probably because it's an accidental invasion and then it's a end of the road for them and the macrophage,
Hmm.
right? Or do they have a mechanism to escape the macrophage?
Well, what I understand is when they get phagocytosed by these macrophages, uh, they can escape from those vacuoles of macrophages and activate the apoptosis of the macrophages, but I'm not too sure whether or not the fate of the bacteria will be the same as the macrophages or not.
Mm. It's interesting because it's very similar to the enteroinvasive E. coli um, that we covered last week. So that enteroinvasive E. coli can go into the enterocyte, and sometimes they come out the other way in the laminocyte, and then they can invade macrophages, but they can escape macrophage to go back into enterocytes.
Ah, well, most likely it would be similar, I would
Mhm.
imagine. Yeah, because macrophages are not the natural target of these pathogens, this Shigella pathogen.
Mhm. And the EIEC also make this actin tail, the rocket-like thing behind them to move
Oh,
around too.
really?
Yeah, they do.
Wow. Okay. So, well, before we get into these actin tails here, the third cell that Shigella can infect are enterocytes. And this is not directly because it does not have specific receptors there, but it does so by first injecting these antigens called invasion plasmid antigens, right? IPA. Injects these antigens by using this injection system, which looks like a syringe here. So you can imagine that little stick here carries a syringe, and then it will just plunge its syringe to the cells, injecting these toxins, I guess, well, antigens there. And what it does there, it changes or reorganizes the cytoskeleton, which makes the cell engulfs and internalize the Shigella inside the cytoplasm. And then once inside, what it does is it escapes the vacuole. So it's just basically tricking the cells so it can get into the cells. Now, once entering the cell, it can control polymerizations of actin monomers. Well, because one of the E. coli, the EHEC,
EIEC,
EIEC, okay.
enteroinvasive one.
Oh, okay. So wait, the invasive one, does it have a flagella?
No, it doesn't have a flagella. It
Ah,
forms this rocket fire thing with the host actin filaments.
okay. Well, similar to that invasive E. coli there, Shigella also generates this actin tails. So what it does is it's manipulating these actin polymers. Later on, it will hijacking and also using it to propel itself throughout the cytoplasm.
What's that vibrant looking picture there?
Ah, so this vibrant looking picture over here is a picture of a cell. Now, I'm not too sure what type of cell here, but what I want to show or remind everyone, students here, is that the cytoskeleton inside the cell is quite complex, quite dense, and not only is it made of actin, but it's also made of microtubules. Now, if you remember microtubules, they're, I guess, the main cytoskeleton when there's mitosis happening, so pulling up those chromosomes. But in a non-mitotic cell, it uses as a structure where it can basically hold mitochondria together or hold organelles in place. It will be the railways of these small kinesins, kinesins, and so on, right? So it's pretty hectic inside the cell. Now, that would be a challenge for these Shigellas to navigate, right? So they're quite cool that they do have, or they do produce these enzymes that can cut off these microtubules. This is also quite interesting. Like this one microbiologist called this strategy as bushwhacking through the microtubule jungle. So you can see there, imagine there, this our little Shig, not only is writing our actin polymers, but also cutting off all the microtubules inside the cell.
Mm. So they have this pirate sword looking thing. So cool.
Oh
So what
yeah,
happens to the what? Sorry.
they're
Yeah,
little pirates.
the little pirates.
Not only they
Even the
hijack.
name Shig sounds like a pirate name. Yeah, and they hijack our cells, right?
Mhm. Well,
So
not only hijack. Oh, sorry. Yeah, there you go.
that bushwhacking thing that they do, they must be inflicting a lot of damage to the cell then, right?
Hmm.
Will the cell be able to survive a Shigella invasion?
Yeah, I'm not too sure, but most likely what I've read is that Shigella invasion will generate into the lyse of the cell, the infected cell itself.
Okay. Well, what do they want to do inside the cell actually? Why do they want to come in and bushwhack their way through a cell? Do they replicate inside the cell?
They're looking for pirate treasure. Um,
Ah, makes a lot of sense.
yes. So
Gold
they
coins, right?
Yeah, well, gold coins and the currency of, I guess, cellular parts, or what not cellular parts, but um,
And what, glucose,
in-
amino acids?
yeah, so amino acids, everything that is needed for bacterial replications, because they do
Lactose. Wait,
replicate.
they're non-lactose fermenting. What
Well,
did you
basically everything.
do? Okay.
Now, what's worse is that they will not only destroy this one cell, but they can destroy other cells. This is quite funny in itself. So eventually, the bacteria will encounter the membrane, right? And most of the time, they will bounce off like a, a ball there, a basketball, I guess, on a wall. But some will try and still penetrate these membranes. And for successful invasion, they will generate the finger-like projections, which eventually will pinch off, generating into this Shigella inside a double membrane uh, layer there, which later on be lyse, and then that means the Shigella will free itself and invade the neighboring cells. Now, the cells by cell extension will basically destroy enterocytes, the group of enterocytes there, and create focal ulcers inside our GI tract. Right, clinical manifestations of Shigella infections, which we also can call shigellosis, is basically diarrhea, fever, stomach pain, feeling need to pass stool. Probably one thing to underline here is that diarrhea can also be bloody and also could involve pus, I think, if I'm not mistaken, because almost purely it's from inflammations um, due to, well, I guess the Shigella itself invading other cells, making those ulcers, and infecting macrophages and so on. Yeah, shigellosis resolves spontaneously, so most likely will not need extensive treatment, but mortality rate is quite high, especially in underdeveloped countries or countries that has, I guess, low sanitation because it's going to be much easier for them to transmit as well.
So the fact that they resolve spontaneously after 2 to 5 days, um, would mean that we would most likely manage to, what do you call this, clear them off just by our immune system alone.
Hmm. Yeah,
It's
um,
nice.
Yeah, that's nice. I'm not sure why though, because they're quite invasive towards these enterocytes.
And your macrophages as well, right?
Well, yeah, they can induce apoptosis of macrophages. So most likely resolving this would need the help of T cells or B cells. Well, most likely would be T cells, I guess. So cell-mediated adaptive immune system there. So it will, I guess, eradicate the infected enterocytes, and B cells will generate antibodies against these Shigella uh, strains that are infecting the body.
If we're relying on our adaptive immune system for this, would that mean that we'll be resistant to further Shigella infections? Like, can you be infected repeatedly with this Shigella if you already had one before? Because it's T
Mm,
cells and B cells, um, and you have some memory.
yes, yes, yes.
Like,
Yes, so memory immune cells, yes, they might offer some protections, but this is also one of the reasons why we rarely see vaccines for bacteria,
mhm.
is because antigenic shifts in bacterias. So they can uh, basically change their antigens, their O, mostly would be O antigens, the LPS there. So later on, it will not be recognized by the memory B or T cells from
Uh-huh.
previous infections.
Okay. Well, they're called Shigella. Do they also produce the Shiga toxin?
Ah, yes, I did not put that in here. I forgot about that. Yes, they do produce the cetotoxin, and basically the cetotoxin that you have covered in E. coli, the EHEC one, right? If I'm not
Mhm.
mistaken.
Yep.
Um, it came from the Shigella dysenteriae, which is the I guess that's the founding species of the cetotoxin.
Hmm. Well, it's been said that the EHAC strain would have acquired the sugar toxin by the action of bacteriophage, which
Mm.
infects Shigella first and picks up the Shiga toxin gene and injects it into E. coli to then make the EHEC strain. And that's interesting because that would mean that, in E. coli, the strain or the serotype that produces Shiga toxin is usually different from the entero-invasive one. But in Shigella, it can do both, right? That means it produces the toxin and it's able to invade the enterocytes. So this is double weapon for them.
Yes, yes, it's a double weapon. But yeah, as what we know about bacteria is like even though they have these all these lists of weapons, not all bacteria strains carry all these weapons. So it depends on the strain that is infecting them.
Is Shigella as versatile as E. coli in terms of picking up virulence factor genes from the environment?
Hmm.
Do they take up plasmids, for example, random strands of DNA as
Uh,
easily as E. coli?
I'm not too sure myself, but from the literature that I've read, I've never read anything about that.
Yeah, probably not. Otherwise, we'll be using Shigella as well as E. coli in our like lab experiments and stuff, right?
Yeah.
Okay.
I think- Yeah. Okay. Oh, I thought I was done there, but identification. I guess identification is would be similar to Salmonella as well as the Enterobacter that we've covered before. We can use Macron-Kieger, well, basically not only Macron-Kieger, but selective media that only can grow Shigella or later on Salmonella. Additionally, biochemical tests, as you can see in this table here, there's multiple tests that can be used to confirm Shigella strains in your samples. Agglutination tests can also be used. This is in response to the O group specific antiserum, so A, B, C, D, the different species or serotypes of Shigella itself.
What would be the importance of identifying a different serotype or um, species there? Do we have different treatments against each one?
As far as I know, the treatments would be the same. It's just that the dysentery, the serotype A there, that's the only species that produce Shiga toxins.
Ah, okay.
And yeah, treatment, usually it's self-limiting. So antibiotic is only used if it is needed. And for prevention, it's basically standard sanitation practices. Oh, also, this is quite nice info here. Flies can serve as passive vectors. So we do not want flies anywhere, right? Even in our office cubicles. Yes.
So we can't really keep flies as pets, not anymore. Well, if they don't get exposed to sewage, they're
Yes,
probably fine.
I guess in a control environment inside the lab or inside a classroom that doesn't have any windows or openings, then sure.
Yeah, as long as you make sure it doesn't go to the toilet when you go.
Okay, I think that's all for my sides on Sigella.
Cool. Well, let's take a break then before we move on to the next one, which will be Salmonella, right?